Hey everyone, and welcome to our laser-focused review for the BIMM 100 final. Today, we're going to absolutely nail the key concepts from Lecture 15 on high-throughput gene expression. Okay, let's just dive right in. This is a huge topic, and you are definitely going to see it on the exam, so let's make sure you're totally prepared. So here's our game plan. First, we'll cover why we even need these techniques. Then we'll walk through the whole microarray workflow and break down a real homework problem you've already seen. After that, we'll check out its more modern cousin, RNA-seq, and finally we'll wrap it all up with the absolute must-know points for the final exam. Okay, first things first. Let's really set the stage for the big problem that these amazing techniques were invented to solve. I mean, think about it. Every single cell has the same genetic blueprint, the same DNA. So what's the deal? Well, it all comes down to gene regulation. Different cells are reading different chapters of that book, and that unique set of all the RNA transcripts in a cell. That's called the transcriptome. It's the key to understanding what a cell is actually doing at any given moment. And get this, we're not talking about just a handful of genes. We've got over 20,000 of them in the human genome. So to really get the full picture of a cell, you can't just look at one or two genes. You need a way to listen in on all of those conversations at the exact same time. And that, right there, is the challenge. So how do we tackle this massive problem? Let's jump into the first major solution we're going to cover, the DNA microarray. The best way to think about a microarray is like a massive, super organized fishing trip. Each tiny little spot on this glass slide has a specific piece of DNA bait, or a probe, that's designed to catch exactly one type of RNA transcript. So by seeing what gets stuck to each spot, we can see which genes are turned on and by how much. Okay, pay attention, because this workflow, it's pretty much a guaranteed topic for the exam. So here's how it goes. First, you take your two samples, let's say cancer cells and healthy cells. You isolate all the RNA. But RNA is kind of flimsy, so you convert it into a tougher DNA copy, called cDNA. Here's the cool part. You label the cDNA from one sample with a red fluorescent dye and the other with a green one. Then you mix them all together and wash that mix over your microarray chip. If a piece of cDNA finds its matching DNA probe on the slide, it'll stick or hybridize, and that spot will light up. And this is where it all comes together. The colors are basically your secret decoder ring for any microarray data Professor Pasquinelli throws at you. If a spot is glowing bright red, it means that gene was way more active in your first sample. If it's green, it was more active in the second sample. What about yellow? Well, that's just red and green mixed, so it means the gene was expressed at about the same level in both. And if a spot is black or dark, simple. The gene wasn't really turned on in either of them. All right, theory is great, but let's actually apply this. We're going to break down a classic exam-style question, one you've already seen on your homework, so this should look familiar. You're going to see an image just like this on the final, I promise. This one is straight out of Homework 8. So what are we looking at? We have a bunch of genes, A through F, and we see how their expression changes when exposed to two different strains of hepatitis B. The question asks us to focus on strain 1, which is the one known to cause cancer, and explain why it's so dangerous based on this data. So what's the very first thing you do when you see this? Don't panic. Just translate the colors into simple data. For strain 1, we can see that genes A, D, E, and F are all red. So what does that mean? They're upregulated when the virus is around. Gene C is green, so that one's downregulated, and gene B is yellow, so no real change there. Simple as that. And here it is. This is the pro tip. The key piece of logic you absolutely need to connect, and it comes straight from the homework answer key. When a gene's expression is changed by something that causes cancer, we can make a really good guess about its job. If it's upregulated, it's probably a proto-oncogene. If it's downregulated, it's likely a tumor suppressor. And this slide just nails the logic, doesn't it? It's all about the gas and the brakes of the cell. Proto-oncogenes are like the gas pedal. They tell the cell to grow and divide. So, upregulating them is like flooring it. Tumor suppressors, on the other hand, are the brakes. They tell the cell to stop. So downregulating them is like cutting the brake lines. Either way, you're heading for a crash. Uncontrolled growth, which is exactly what cancer is. Okay, so microarrays are super useful. But now let's talk about the next generation, the real modern powerhouse, RNA sequencing, or RNA-seq. This takes a totally different approach. The big difference with RNA-seq is we're not just asking if a gene is on. We're literally reading the genetic messages themselves. So you still start by getting the RNA and making cDNA, but instead of just seeing what sticks to a chip, you chop up all the cDNA and feed it into a super high-powered sequencing machine. This machine reads the actual genetic code, the A's, T's, C's, and G's, of millions of these little fragments. It's like instead of just checking a book's table of contents, you're reading every single word on every page. Okay, this slide is huge for the exam. The microarray versus RNA-seq showdown. Here's the bottom line. A microarray is like a dictionary. It's great, but it can only tell you about the words you already know exist, the genes you put probes for on the chip. RNA-seq, though, is like having a full conversation. It can tell you about words you've never even heard before. It can discover totally new genes, find tiny typos or mutations, and even show you different versions or splice variants of the same gene. It's a genuine discovery tool. All right, we are in the home stretch. Let's boil all of this down to the absolute must-know critical takeaways for the final. Okay, this is your final mental checklist. Before you walk into that exam, make sure you can do these 4 things. 1. Explain the main goal, that both of these techniques measure gene expression across the whole genome. 2. Be able to instantly translate those microarray colors. Red, green, yellow, black. Know what they mean. 3. Apply that cancer logic. Upregulated is a proto-oncogene, downregulated is a tumor suppressor. And 4. Clearly explain that fundamental difference. Microarrays confirm what you know, but RNA-seq can discover what you don't. And just remember, this stuff isn't just about passing a test. These are the real tools that scientists are using right now to make incredible, life-saving discoveries in medicine. Understanding this is understanding the language of modern biology. Look, you've put in the work, you get these concepts. So just walk into that exam, trust what you know, apply it with confidence, and you're going to totally crush this final. Good luck.